Ding away.
Hi. Oh, hey, Scott.
Hi there.
Oh, very nice to meet you.
I apologize, I have to run out.
Oh, that's fine.
We should chat at some point. But.
Oh, hey, Kate.
I grew up in Castro Valley.
Oh, okay. Very nice to meet you.
Hey everybody, thanks very much
Okay.
for coming out.
Okay, yeah, no problem. Nice to meet you.
So we can move on here. So I want to thank Dr. Tyler Evans for coming out today. He's our scheduled speaker cancelled, so I called out one of my markers up there at Cal State East Bay and got Tyler to come down. His lab at Hayward is focused on researching how shifts in abiotic variables affect the performance of marine organisms, particularly within the scope of global climate change. The environmental factors his lab have been studying so far include increased temperatures, fluctuating salinity, and decreases in ocean pH. Tyler's been at East Bay since 2012. He has his Bachelor's of Science and his PhD from the University of Saskatchewan, so he's a Canadian. And
I'm still here.
he's still here. He hasn't left yet, but who knows what happens in the next 4 years.
My passport is up to date.
He did postdocs at Hopkins Marine Station with George Summerall. He had another postdoc at the Fisheries and Oceans Canada and also at UC Santa Barbara with Gretchen Hoffman. He's here today to talk to you about the role for transcriptomics in environmental physiology, global change biology, and finding the genes that matter for environmental adaptation. So let's give a Mars landing welcome to Tyler Evans. Thank you.
Well, thank you so much for having me and hosting me today. Always happy to come out and do these things, especially in such a wonderful location, right? So as the title of my talk alludes to here, what I'm going to be talking about today is mostly about this technique called transcriptomics. All right, so I thought I should at least briefly define transcriptomics before I get into the data. So when I say transcriptomics, what I'm talking about is the study of the transcriptome. All right, and then what the transcriptome is a complete set of RNA transcripts, so mRNA, all right, produced by the genome, all right, under specific circumstances or in a specific cell, all right, and we usually use these so-called high-throughput methods, all right, to detect these changes in mRNA abundance. Used to be microarrays, all right, these days we're almost exclusively using RNA sequencing to find that out. All right, but the point is what transcriptomics does, it allows the identification of RNAs whose abundance is shifting, all right, in response to some kind of change in the conditions. And then what I'm going to talk about is mostly about changes in the environment and how that affects the expression of all those RNAs. All right, and I want to do it, talk about transcriptomics, how the transcriptome is affected by the environment from a couple different perspectives. All right, so the first part of my talk, all right, what I want to talk about is sort of showcase transcriptomics as a tool to address problems in environmental physiology and problems in global change biology. All right, but then in the second part of my talk, I want to sort of have a broader discussion, all right, about transcriptomics as a tool. So more of sort of the approaches as transcriptomics, all right, and in particular, the role of transcriptomics in identifying the genes that matter for environmental adaptation. All right, but for the first part of my talk, we'll focus on some applications that we can use transcriptomics to inform questions around environmental physiology and global change biology. All right, so I want to focus on a case study here that has to do with climate change physiology and some of the work that I've done in ocean acidification. All right, and looking how ocean acidification affects these purple sea urchins, which of course are found in the California current, all right, right on the west coast of the United States. All right, so we're dealing with a marine audience here. So most people are probably familiar with what ocean acidification is. All right, but briefly, all right, it's a direct result of atmospheric CO2 emissions from humans. All right, and as humans release all that CO2 from things like burning fossil fuels, deforestation, things like that, all right, it accumulates in the atmosphere. And that's what's shown in this red line here. And you can see through time, you can see this accumulation of carbon dioxide in the atmosphere. All right, about a third to a half of that atmospheric carbon dioxide ends up being dissolved into the global ocean. All right, so as CO2 increases in the atmosphere, there's a corresponding increase in CO2 concentration, all right, in seawater, and that's what's shown in this green line here. All right, as that CO2 gets dissolved into seawater, all right, it undergoes chemical reactions in seawater, and it actually changes the chemistry of our world's oceans. All right, in this slide, I've shown a couple of the more important chemical changes, or at least the chemical changes that are most relevant to things that live in the ocean. All right, so one of these sets of chemical reactions caused by CO2 entering the ocean serves to reduce the pH of the ocean. That's why it's called ocean acidification. All right, it occurs as carbon dioxide reacts with the water in seawater, producing carbonic acid, and then dissociating into bicarbonate and hydrogen ion. All right, and of course, it's this increase in the concentration of hydrogen ion that reduces the pH. All right, another important set of chemical reactions that occurs as a consequence of all this atmospheric carbon dioxide, all right, is on the lower half of this slide. All right, and those chemical reactions consume carbonate. In other words, it removes some of the carbonate that's in the ocean. This occurs when hydrogen reacts with the carbonate in the ocean to form even more bicarbonate. All right, and of course, where carbonate comes in, or that's sort of the building block that marine calcifiers use to make their hard parts, to make things like shells, skeletons, and spines. All right, so by having less of this carbonate available, all right, it's hypothesized, and there's been, at this point, a really strong literature showing that these marine calcifiers that are making their hard parts from calcium carbonate are having a harder time doing so because there's less carbonate available. All right, as we've sort of continued to study and discuss the effects of ocean acidification on marine organisms, all right, and we've been studying this phenomenon for some time now, I think one of the more recent developments, one of the more recent key findings, all right, is what's written at the top of this slide here. All right, it's that the biological consequences of ocean acidification are likely to manifest regionally. All right, and the reason for that is because not all marine populations currently experience the same pH condition. In other words, there's a lot more pH variation, natural variation in the ocean than we originally sort of predicted or that we originally thought. All right, and an ecosystem that really emphasizes that trend, all right, is right outside our door in the California current. All right, so this is an ecosystem of really high pH variation. All right, and the reason that it exhibits this pH variation, all right, is because it's a coastal upwelling system. All right, what upwelling is the result of a seasonal shift in wind direction. All right, and what that seasonal shift in wind direction does is it blows surface waters offshore. All right, and as a result of those surface waters blowing offshore, deeper, colder water, all right, moves up to take its place, all right, or is upwelled. All right, and importantly, those upwelled waters are high in CO2, they're high in carbon dioxide, all right, which makes them low in pH and also reduces the concentration of carbonate. All right, and you can see that these chemical changes that are associated with upwelling, they're exactly the same chemical changes that are occurring with anthropogenic, atmospheric CO2 emissions and ocean acidification. All right, a couple of important points about that upwelling and how that affects pH variability, all right, is that that variation can be quite large. All right, and the other thing that's important to consider is that variation can occur in biodiverse regions. All right, so for example, the intertidal, that's what's shown in this graph here. So this is some pH data, all right, that was collected from autonomous sensors placed in an intertidal region in coastal Oregon, a place called Fogarty Creek. All right, so we have time on the x-axis and then pH on the y-axis. All right, what you're looking at is a 3-day running mean in blue. All right, but you can immediately appreciate that the pH is quite variable. All right, there's in the green line, we see the pH minimum for this site in Fogarty Creek. It's somewhere around pH 7.5. All right, to put that into perspective, all right, that red line there, that's the average pH in the global ocean right now. All right, and this shaded box here then shows the predicted pH for the year 2100 in the global ocean. All right, so you can see that this area of high upwelling that we see in Fogarty Creek, all right, is already exposing organisms to pH conditions that we wouldn't expect to see in other parts of the ocean for several decades into the future. All right, not until the year 2100, for example, in the open ocean. All right, to sort of complicate things even further, all right, the influence of upwelling, all right, is not consistent across the entire California current. All right, so what I showed you in Fogarty Creek, that's an area of particularly strong upwelling on the Oregon coast. All right, but not all locations along the California current experience that upwelling in the same degree. All right, and that's what's sort of shown in this diagram here. All right, so we've placed autonomous pH sensors in intertidal sites all across the California current. All right, the red dot showing the site in Fogarty Creek that I showed you previously. All right, but we have other sites where we have these same sensors placed in Oregon, all right, in Central and Southern California. And what's this shown, all right, is that they're not all experiencing the same pH regimes. All right, a place like Fogarty Creek is going to be exposed to low pH conditions much more frequently, all right, than a place like Alegria, which is down in Santa Barbara in Southern California. All right, so what's shown in this graph here is the frequency of those measurements from those autonomous sensors that fell below pH 7.8. All right, you can see in this red dot, that's from Fogarty Creek. All right, about 20% of the measurements are actually below pH 7.8. If we compare that down to sites in Southern California, like Alegria here, all right, it's only about 1 or 2% of all the measurements that actually fall below that threshold. All right, so marine populations in Fogarty Creek are encountering low pH conditions much, much more frequently than populations down in Southern California. All right, so this ends up with this sort of pH mosaic, right, where we have these different local pH conditions across the California current. All right, and the existence of that mosaic brings to mind a number of important research questions, and that's what's shown on this slide here. All right, so these are some of the questions that I hope to address in some of the research that I've been doing. All right, one of those questions is, do responses to seawater acidification differ across species ranges? All right, secondly, what mechanisms allow marine organisms to tolerate low pH seawater? So if we have a place like Fogarty Creek, for example, where we know animals living in that environment are already experiencing low pH conditions lower than elsewhere, all right, we might be able to figure out how they're doing it. We might be able to figure out the mechanisms that are allowing them to survive in those low pH environments. All right, and then finally, are organisms frequently exposed to low pH seawater more resistant towards future acidification? So in other words, does the ability to cope with pH variation today somehow confer resistance or make them more tolerant to deal with future anthropogenic ocean acidification? All right, the model system that I use to address all three of those research questions, all right, is early life stages of the purple sea urchin. All right, and this is a really great, really excellent model system for a number of reasons. All right, first, it's distributed across that entire pH mosaic in the California current, so it's found, the same species is found from southeast Alaska all the way down to Baja, Mexico. All right, they're marine calcifiers, including their early embryonic and larval life stages, so they're using carbonate to build skeletons during that time. All right, in terms of the genomic approaches that I was interested in taking, all right, these purple sea urchins have a fully sequenced genome, which makes doing those things a lot easier. All right, and then finally, in recent years, there's been evidence for local adaptation to pH. In other words, we've some studies that have seen changes in allele frequency between sea urchin populations that suggest there's actually selection for more tolerant pH genotypes. All right, so there's some evidence for local adaptation to pH. All right, so here's a look at the experimental design that I used to address those research questions that I showed you. All right, again, we're interested in finding out population-specific transcriptomic responses to seawater acidification. So we want to see how gene expression varies between those different populations. All right, and so here's the experiment that we used to address those questions. We took adult urchins from each of those 6 locations where we had the pH data. All right, we collected them from intertidal sites that were very close to the sensors. All right, we took them back to the lab. All right, and we bred the males and females. All right, and then we raised the progeny under 2 different conditions. All right, one sort of lower CO2 condition, about 385 microatmospheres. That would be the equivalent of sort of present-day PCO2 conditions, at least those in between upwelling events. All right, and then a second treatment, a high PCO2 treatment at 1,000 microatmospheres, which would be representative of a typical upwelling event, all right, or some time in the future between upwelling events. All right, we then allowed those embryos to grow under those conditions, all right, for about 24 hours or until they hit this stage. This is called the gastrula stage. All right, it's an early embryonic version of the sea urchin. All right, then we extracted the RNA from those gastrula and used Illumina RNA sequencing to look at the changes in gene expression. All right, we took a particular approach. We used pattern matching software. All right, and we told that software to pull out genes that were either strongly correlated or whose expression pattern was strongly correlated or strongly anti-correlated to the frequency of exposure to low pH in the natural environment. All right, and so what I mean by that is that software pulled out genes that were expressed very highly, all right, in populations like Fogarty Creek that would experience low pH conditions often, and lower in the sites in Southern California, all right, or the opposite of that. So very low in Fogarty Creek and comparatively higher in Alegria or the sites in Southern California. All right, and so this is what's called a heat map of those gene patterns that I just described. All right, so in each column, you can see the different populations that we sampled. All right, and then each row of that heat map is the expression pattern for a particular gene. All right, the yellow meaning that gene was highly expressed, blue meaning it was expressed at lower levels. All right, and you can see that our approach basically pulled out 2 sets of genes, all right, genes that were highly expressed, all right, in the populations that see low pH most often, all right, and then lower in those other populations, all right, and then of course the exact opposite trend. So low in the populations that were more frequently exposed to low pH and higher in the Southern California populations. And there's a couple sample expression patterns just to show you that result even further. All right, so I want to focus on this particular set of genes here. All right, so the ones that were expressed at highest levels in the populations that encounter low pH most often in the wild. All right, when we look at the numbers and we look at the types of genes among that gene set that was upregulated in those populations, all right, we see a very strong signature of ATP production. All right, so we see genes changing, all right, that are involved in multiple, all right, major ATP producing pathways, all right, in these systems. All right, and that's what we're seeing here in this chart. All right, so these are pathways, or what's called ontologies or gene functions, all right, where we see lots of genes from those particular pathways or functions. All right, you can see things like the tricarboxylic acid cycle, aerobic respiration, fatty acid, all right, respiratory chain, the mitochondria. All right, these are sort of the major energy-producing pathways that these urchins use. All right, and again, these are in the populations that are most frequently exposed to low pH. All right, just to drive that pattern home a little bit further, here's a diagram showing the citric acid cycle. All right, and the populations that were most frequently exposed to low pH in the wild had higher levels of expression for the first 5 reactions in the citric acid cycle. So those enzymes here that are catalyzing the first 5 steps in the citric acid cycle. All right, so we saw this very clear, very obvious pattern of genes that suggests that these urchins are making more ATP. They're using more energy when they're kept under low pH conditions. All right, and what we think is happening, or what at least we hypothesize, all right, is all that extra ATP is going to maintain pH homeostasis and maintain skeletogenesis. So basically make sure that they can still continue to build their skeletons. All right, we have some other evidence for that. All right, so this is a look at the spicule of the urchin larvae. All right, so this gray part, that would be the calcium carbonate part. All right, it's surrounded by this syncytium. All right, and we know that the inside between the syncytium and the skeleton, that the pH around that area remains high. All right, so despite that the extracellular environment, all right, or the ocean has low pH, all right, urchins work to make sure that this area around the site of skeletogenesis remains high. All right, in order to do that, they have to pump hydrogen ions out, and they have to pump buffer in. All right, that's an energetically expensive process. That requires a lot of ATP to maintain that high pH level. All right, we also know that in terms of calcification, these urchins see very little impact on calcification, even when they're cultured under low pH conditions. All right, so it's suggesting that they can compensate for these changes, but what we're seeing is the ability to compensate comes at an energetic cost. All right, they're simply devoting more ATP towards pH regulation. All right, and this is showing data from another group. All right, this isn't our data, but it's from another group. All right, showing that energetic cost that comes with living in low pH seawater. All right, so this is some pie charts showing ATP consumption in purple sea urchin embryos and larvae. All right, so here's ATP consumption under ambient conditions. All right, and here's a higher PCO2, low pH conditions. All right, the gray pies are showing ATP devoted to ion transport. The black pies showing ATP devoted to protein synthesis, and the white pie is showing what's left over after accounting for those two processes. All right, what you can appreciate if we focus in on the white pies, all right, you can see that under the high CO2 condition, that white pie gets smaller and smaller. It means they're having to use more ATP for protein synthesis and for ion transport, likely related to pH regulation. And it means that there's going to be less ATP left over to do other things. So to reproduce, all right, for behavior, or also importantly to respond to other stresses, to respond to things like ocean warming or ocean hypoxia. All right, so we think what we're seeing is this energetic trade-off associated with increasing pH tolerance. All right, so if we go back to our research questions, all right, we think we have evidence, all right, that there are populations that are more frequently exposed to pH react differently, all right, than populations that aren't as frequently exposed to low pH conditions. All right, in terms of the mechanisms that allow them to do that, all right, we have evidence showing that they're basically using additional ATP resources for acid-base regulation. All right, and then final question, are those urchins inhabiting areas of low pH more resistant towards future acidification? We don't really know, but our evidence suggests that they're going to have less energy to cope with additional changes in the environment. All right, so they may be more tolerant of present-day conditions, but it doesn't necessarily mean they're going to be more tolerant of future conditions. Okay, so I wanted to go through another sort of case study and how we can use transcriptomics to address questions relevant to environmental physiology. All right, in this case, I want to talk about salmon. I want to talk about how transcriptomics can be used to inform conservation. All right, so this was work that I did at Fisheries and Oceans Canada. All right, trying to use these transcriptomic tools to inform the conservation of salmon. In particular, what we were trying to accomplish, all right, is find spatial and temporal patterns of physiological change as these salmon were migrating, all right, on their spawning migrations. All right, so we focused on a particularly vulnerable life stage, or at least it was vulnerable at the time. All right, so these were sockeye salmon. All right, and these were sockeye salmon that were undergoing these reproductive migrations. All right, so here's a look at the salmon life cycle. All right, but they start their life off in freshwater. They'll then migrate to the ocean for 3 or 4 years, and then of course they migrate back into freshwater in order to reproduce. All right, so we were studying this phase of the life cycle right here, these adult spawning migrations. All right, in the case of the sockeye salmon that we were looking at, all right, they were migrating from southeast Alaska, all right, down here, all right, to the Washington, British Columbia border. This is where the Fraser River is. This is the largest sockeye producing river in the world. All right, and there's the mouth of the Fraser River, and then of course they were migrating upstream, all right, through these rivers and streams to get to their spawning grounds where they lay eggs and reproduce. All right, the reason that we were focusing in particular on sort of this life stage, all right, is that migrating sockeye salmon are important for a number of fairly obvious reasons. All right, in terms of ecological importance, all right, they're considered keystone species in marine and freshwater, all right, and also riparian ecosystems. All right, they're very economically important. The commercial and recreational fisheries are a $1 billion industry. All right, these are resources that are shared between the United States and Canada. All right, so in terms of jobs, we're talking tens of thousands of plus jobs in the Pacific Northwest. All right, and then there's also cultural importance to consider. All right, so the indigenous, the First Nations people that live in these regions and traditionally hunted these salmon, all right, the salmon are integral to their mythology, all right, their spiritual integrity of those populations that are found on the West Coast. All right, so given this very sort of obvious importance for sockeye salmon, all right, when there becomes fewer of them, people take notice. All right, there's very obvious, very clear consequences. All right, and unfortunately, that's what we've seen over the last few decades is sort of these diminishing returns of sockeye salmon in the Fraser River. What you're seeing here is a graph of the number of returning fish over time. That's what's on the x-axis here. All right, and you can see, especially in recent years, we've seen a very sharp decline in the number of returning sockeye salmon. All right, so you'll notice in the last about 20 years or so, we've only had 2 or 3 years where it's actually been above the historical average. Most years, it's below that historical average. All right, of course, that has consequences that reverberate through all those different factors of importance that I talked about in the previous slide. All right, you'll notice this dot right here. All right, that was the year 2009. All right, that was a year of particularly severe mortality. All right, it was characterized by what is called marine mortality rates. It means that a lot of salmon left the river and made it to the ocean. All right, but very few actually came back to spawn. All right, and when I say very few, all right, I mean the difference in this graph right here. You probably don't need a graph to tell you the difference between 10 million and 2 million. All right, but nonetheless, based on the number of salmon that left the river as juveniles, they expected about 10 million to return as spawners. Just under 2 million actually came back. All right, so needless to say, there was a lot of people, fisheries and oceans, fisher people, thing like that, wondering exactly where these 8 million some odd salmon went. All right, so one of the things that fisheries and oceans was interested in using to try and solve this problem, all right, was to figure out, well, maybe transcriptomics, all right, and maybe we can look at the transcriptomes of these migrating salmon, and that can provide clues that will help inform their management. All right, so these are some of the research questions that I tried to address using transcriptomics. All right, firstly, can salmon transcriptomics be used to predict survival during migration. So in other words, can we look at the transcriptome, and does that give us hints as to whether or not this is going to be a salmon that's going to successfully reproduce, or is this going to be a salmon that's going to die before it actually gets to the spawning grounds? All right, and then can those transcriptomes point towards a cause of death, right? Obviously, if you know what's killing the salmon along those migration routes, it makes conservation much easier. All right, and then finally, is the expression of particular genes indicative of migration success or failure? In other words, can we use individual genes to predict fate? Do we have these biomarkers of fate that can be used to predict whether or not a fish is going to be a successful migrant or a premature mortality? All right, so this is a look at what I think is a really innovative, really creative strategy that integrates transcriptomics to address some of those questions. All right, so what we did is that we collected wild adult salmon. All right, we intercepted them along their migration route. So we started right down here where this green arrow is, all right, right in the river mouth of the Fraser River, and we captured these adult migrating salmon along their journey. All right, when we captured them, we took a few pieces of information. All right, one of the first things we did is that we inserted a radio tag into their stomach, and we used that radio tag for biotelemetry. We used it to track their progress through the river. All right, we were able to do this because there are these receiving stations set up along the river. That's what's indicated by these red dots here. These are different receiver stations. All right, so every time a fish would swim by one of these receiver stations that had this radio tag in its stomach, it would send a signal to say that the fish made it this far, right? And so you could track the progress of the fish as it migrated upstream. All right, the other piece of information we took from these fish was a fin clip for DNA genetic stock ID. All right, because these fish are reproducing in the same spot year after year, their populations are very structured. In terms of their genetic background, they're completely distinct. All right, and so importantly, what that allows us to do is it allows us to tell us where the fish are going. All right, so we can use the DNA to tell us which particular population those salmon belong to, and that can tell us where the fish are intending to go. All right, and we combine those two pieces of information, you basically have an equation to get survival. All right, so if we deal with, for example, this quenelle population, all right, if we have a fish where we get signals all the way along this route, including this one very close to the spawning grounds, all right, we can be fairly confident that it's successfully migrated to the spawning grounds. All right, if we get signals for some of these receiver stations, but not the one closest to the spawning grounds, it's probably a premature mortality, and it didn't make it all the way to the spawning grounds. All right, the last thing we collected was a non-lethal gill biopsy, so a little clip of gill tissue, and we can use that for transcriptomics. So we looked at transcriptomics. In this case, we used a microarray. All right, but what that allowed us to do is allowed us to look at differences in gene expression between successful migrants and those that didn't make it. All right, and this is a look at some of the genes that we saw that were differentially expressed between successful migrants and the premature mortalities. All right, so this is, again, another one of those heat maps where you have the individual fish in the columns and genes in the rows. This time, the red means the genes that are upregulated and green, the genes that were downregulated. All right, and you can largely see that there's opposing expression patterns between the successful migrants and the premature mortalities. This is the expression pattern for the premature mortalities here in these orange bars. All right, and you see a largely different set of expression for those genes in the migrants that were actually successful in making it. All right, we also looked for specific genes to see if there was just maybe one or two genes from that list that could actually be used to predict fate. And we found this one. All right, it's called Cofactor required for SP1 transcriptional activation. It doesn't really matter what it's called, right? It just matters if it can predict fate or not. All right, and what we found is that animals that had lower expression levels of this gene were much more likely to finish their migration. All right, so they're able to survive longer in the river and they're also able to travel farther in the river if they had comparatively lower levels of expression for this gene. All right, I should mention that in this diagram, all right, these genes were enriched for processes related to the immune response and also related to cell death. So we think, all right, that, and there's been other evidence to come out since, all right, that there may be some sort of virus that's infecting these salmon, all right, and that's what's causing this large mortality that we've seen. All right, so to go back to those research questions, can transcriptomics be used to predict survival during migration? Potentially. We certainly found a gene expression pattern correlated with successful migration, but it's yet to be put into any kind of policy. All right, 2, can it point towards causes of death? When we look at those expression patterns that we just saw, all right, the unsuccessful migrants, the premature mortalities, tend to have a higher expression of immune response genes, all right, genes involved in cell death that would be indicative of a viral infection. All right, and we also talked about CRSP1 as this potential biomarker of success or failure. Okay. So that covers what I wanted to talk about in the first part of my talk. For the next 10 minutes or so, I just wanted to talk about a broader discussion about the role of transcriptomics, in particular in identifying the genes that may be most important for environmental adaptation. One of the things that happens when you do these techniques is you get these long lists of genes, all right, and you have to figure out which of these genes, if any of them, are most important, right? And I think there's a general assumption that the genes that are differentially expressed, the genes that we've identified in some of these studies, are important for actually adapting to those conditions that we've talked about, all right, but there's some interesting data that's come out recently that suggests we should maybe think about this a little bit more, all right? So that's what I want to talk about for the next couple of minutes. All right, so I recently had the opportunity to write a review about this topic, and the review really focused on trying to answer this question here. All right, so do the genes differentially expressed during environmental stress have large effects on the fitness under these conditions? So we know genes that are different, lots of genes are being differentially expressed, but which ones are most important? Which ones are actually having an impact on fitness? All right, and actually sort of experimentally addressing that question is really quite challenging, all right, because you have to know all of the genes that are changing in response to a particular stressor. You also need to know the ones that aren't changing in response to the particular stressor. And then you need to know the impacts on fitness of each one of those individual genes. All right, and so you can sort of see from an experimental point of view, that's a very difficult set of experiments to actually perform. All right, but there are a couple of species where we can do these type of experiments. Unfortunately, none of them are in the ocean. All right, but yeast are a very powerful model system where we can do these type of experiments. All right, they have a small genome. All right, so there are very few genes, protein encoding genes in that genome. And what that allows researchers to do is they can systematically knock out every single non-essential gene in the genome. All right, and that's what they've done in this experiment here. All right, so they've taken the wild type. All right, this time they're interested in looking at which genes are important for adapting to temperature stress. All right, so they've taken wild type, they've exposed it to a heat shock. All right, and then they've analyzed that genotype for performance. So they've looked at survival of the yeast and also looking for a change in growth rate. All right, and then they can repeat that for a gene, but knocking out one gene. All right, so they'll knock out one of the genes, and they'll repeat that same process, looking for a difference in survival or growth rate when that gene is knocked out. And they can do that for every, as I said, every non-essential coding gene in the genome. It ends up being 2,954 genes. All right, what they've found, I think, is really quite striking. All right, and this sort of summarizes what they found. The genes important for protection against acute heat stress, all right, they're not the ones that are changing their expression patterns when you expose them to temperature. All right, and that's what's shown in these heat maps here. All right, so if you see bright red or bright green, those are genes that are changing their expression when those yeast are exposed to high temperatures. All right, bright blue or bright yellow means that there was a significant effect on death rate when that gene was deleted. All right, and so you can see lots of red and green, but very little blue or yellow. All right, it basically means that the genes that were changing expression during heat stress, all right, are not the ones that had an effect on death rate when they were actually deleted, at least individually, deleted one at a time. All right, this figure here maybe shows that relationship a little bit closer, a little bit more clearly. Here's gene expression on this axis. All right, here's the change in death rate that occurred when that gene was deleted individually. All right, so 0 here, all right, would indicate that there was no significant change in death rate when that gene was deleted. All right, and what you can see, if we look at, for example, a gene like heat shock protein 30 here, you can see its expression undergoes a huge change, 110-fold change when it's exposed to temperature stress. All right, when you delete HSP30 in yeast, there's no significant effect on death rate. All right, and interestingly, all the ones that did have a big effect on death rate are the ones down here. You can see that death rate increased in all these gene deletions, and you can see there's their fold change. All right, so small fold change, despite the fact when you delete them, they have a big effect on death rate. All right, and this is certainly not the result of just one experiment. All right, so there's been a number of experiments that have shown this in yeast, and again, the major caveat, of course, is how is this going to apply to other organisms? But yeast is really one of the only organisms where we can actually do these types of experiments. We actually have data for it. All right, but you can see some quotes from these reviews that show, generally speaking, the genes that are changing in response to a particular stressor don't appear to be particularly important for actually modifying tolerance limits, at least when they're deleted individually. All right, that pattern even applies to the heat shock proteins. All right, so heat shock proteins, sort of the hallmark of heat stress, right? We get very strong induction of heat shock proteins when we expose animals to high temperature. All right, that's what's shown here. Again, we're looking in yeast. All right, so there's genes that are exposed to high temperature, and you can see very strongly upregulated. All right, but when we look and we start deleting those genes and seeing if they have an effect on death rate, there's only 2 of the 29 heat shock proteins encoded in yeast, all right, that actually have a significant effect on death rate. That's HSP 104, all right, and JJJ2. We see a similar pattern in C. elegans, so a nematode worm, where we can do these types of systems experiments. All right, so in this case, what these researchers did is they looked individually deleted 219 chaperone proteins, so heat shock proteins included as molecular chaperones that are involved in protein folding, and then they looked at the capacity to change protein folding. When those genes were deleted, what they found is only 21 of the 219 deletions actually saw a reduction in the capacity to fold proteins. All right, so what this suggests then is that there's lots of redundancy built into these systems. In other words, you've got 219 heat shock proteins. If you delete one of them, there's 218 left to take its place, and you're probably not going to see a big change in fitness. All right, of course, the important question is, what were the genes that did change? If it wasn't the ones that were undergoing a big expression change, which were the ones that actually did affect growth rate when they were deleted? Well, it's constitutively expressed genes. It means genes that had proteins in the cells. They just didn't alter that abundance when they were exposed to stress. All right, and what are called a few highly connected genes that regulate, or that's the key word, regulate aspects of the stress response. All right, so it wasn't the heat shock proteins that are actually going to sort of fix the problem. All right, it was the regulators, the ones that turn the heat shock proteins on and off, that were disproportionately important. And then remember, those are the ones that didn't change their expression and therefore were not detected by transcriptomics. All right, an example of one of those regulatory proteins, all right, would be kinases and phosphatases. So these are enzymes that can add phosphate groups or remove phosphate groups to proteins. All right, and by doing that, they change protein activity. It doesn't affect their abundance, but it affects their activity. For example, it might open or close an ion channel, for example. But the abundance of that protein is going to remain unchanged. All right, and there were several of these phosphatases or kinases, all right, that had a significant impact on death rate in yeast, all right, when they were deleted. All right, we've done some experiments, or I've done some experiments to look at the role of phosphorylation, to look at the role of adding or removing phosphate groups in response to temperature change in some non-model species. So this is some work that I did in mussels. All right, these two species of mussel, Mytilus galliprovincialis and Mytilus trosselis, both are found along the California coast. Galliprovincialis is an invasive species from the Mediterranean, and as a result, it has a much higher temperature tolerance. So it's a worm adapted with a higher heat tolerance compared to the native species, Mytilus trosselis, which is cold adapted. All right, but what I did is I used a set of antibodies that could bind and could show proteins that had been phosphorylated following heat stress. All right, and that's what's shown in these data down here. All right, so this is a Western blot, so every time you see a dark band on there, that's a protein that has a phosphate attached to it. All right, and you can see here's the two species during acclimation. They're acclimated at 14 degrees Celsius, and you can see some bands indicating some of those proteins are phosphorylated. An hour later, and following a 6-degree increase in temperature, you can see a lot more dark areas, right? It means that all these proteins at these different sizes are being phosphorylated, and potentially their activity is being changed. This was data for a particular group of kinases called Mitogen-Activated protein kinases. All right, but we see the exact same pattern with other kinases as well. So these are cyclin-dependent kinases that control aspects of cell division. Here's protein kinase A that control aspects of cell survival. And you can generally see low expression levels during acclimation. All right, and even just 60 minutes later in a 6-degree shift in temperature, proteins start to be phosphorylated. So we don't know the functional significance of that phosphorylation, all right, but it seems that it's likely going to contribute to the differing thermal tolerances between these two mussels. All right, so in terms of the second part, I think an important take-home message is that differential expression may not equal importance, all right, because individual genes tend to have redundant function. There tend to be another gene around that can compensate for it missing. All right, and these upstream sensing and signal transduction genes seem to be disproportionately important for adapting to stress, all right, but they're much more harder to identify via transcriptomics, so they're not undergoing these big changes in expression. Okay, and with that, I'd like to thank the many people that had a hand in some of the research that we talked about today. All right, so the Omegas group for the Urchin stuff, Fisheries and Ocean Canada for the salmon, and Hopkins Marine Station and the Somro Lab for some of the mussel stuff. And if there's time, I'll be happy to take questions.
So, questions?
Yes, go ahead.
In the heat map, so some of the genes are upregulated and downregulated in a is it quantitative? You mentioned like the factor by which they increase like tenfold or.
Yeah, so we usually do it by the log of the fold change. So it's basically, you know, of how many times more was it expressed in the stressful conditions compared to the non-stressful.
Can you actually budget within the microarray the quantitative number of RNA molecules that went up against the quantitative number of, the grams of RNA that went up versus down to find out what happened to the net?
So the net, right? So you don't know the net. So it could be, you could see, in other words, you could see an mRNA increase abundance not because more is being produced, but because more is being degraded, right? And so those types of questions we can't address. We can just see the shift in abundance, but we don't know if it's due to upregulation or it could be due to having more RNA degraded under a particular part.
I'm just thinking of this as in my geological time scale when we used to think about ways to figure out how to know whether microbes were growing fast or slow. We thought it would be a really good idea to look at RNA to DNA ratios, bulk, just plain old
Right.
bulk of RNA against DNA. And yes and no, it kind of sort of worked, but because they're going up and down, the balance would be shown in, you could actually budget it with the fluorescence intensity of each one of those, each one of the genes you have on an array. Is that correct? In a theoretical
So if we're talking about the microarrays, basically what you're looking for is difference in fluorescence, where one group is labeled with red fluorescence and one is green fluorescence. So when you're saying fold change, you're really just talking about difference in fluorescent levels. Intensity. That's what it measures. When we switched to RNA sequencing, which the Urchin stuff was based on RNA sequencing, it's totally different. It's not at all done that way. You're basically just counting how many small little sequences align to a specific part of the genome, and you're counting how many of those you got, and that gives you the gene expression. So it gives you the same data in the end, but calculated through completely different means.
And it's just the same, and so that was from a single cell view, but in a muscle, does it matter where you clip the tissue?
Yes, absolutely, absolutely. So if you took muscle tissue muscle tissue in the muscles, right, adductor muscle or something like that, or gill tissue or a gonad, you can see different expression patterns because those tissues are doing different things, right?
I don't know where to look.
You hope for the best. So when we looked at in salmon, for example, we looked at the gill tissue. We also took liver and muscle tissue, but the gill is where all the osmoregulation happens, and they're switching salinities. So that's why we thought we'd want to see the gill.
Yeah.
So you're making an intelligent choice, knowing that you can't do everything. But the urchins were in larvae, so there's like 10,000 larvae all combined into one sample. So different in the urchin, but again, none of which is particularly ideal, but you're making educated decisions based on science and, of course, money. Yeah. It's expensive to do this stuff. Yeah.
Give me like what?
So the urchin stuff was we did a lot of sequencing for that, so maybe 30,000.
Well, did you send it out or
Send it out. We don't do the sequencing in house, but you can get it done at UC Berkeley, for example. That's where I do.
Grind, extract, mail.
If you do all that, it's going to add up your costs. So I can do those things in my lab. I can do everything but the sequencing. So the sequencing is still expensive, but I can extract the RNA. I can make the library. It's basically prep it for sequencing. They do the sequencing, and then they send, in this case, 3 terabytes of data back to me to deal with. Yeah.
So with the urchins, you saw changes in transcription and changes in energy budget, but it seems like you didn't see necessarily a obvious change in survivability. Do you think that if you were using set pH conditions for the low and the high, do you think if instead you used kind of dynamic pH where the high pH was just a certain amount lower than ambient and was fluctuating, so you had like really low when it fluctuated low and then more average when it fluctuated high, I think you might have seen some more survivability changes?
So we didn't monitor survival, so we don't know. We know that the density doesn't change. The density in the cultures doesn't change too much. So we don't think that there's massive amounts of death, and we know from looking at their morphology that they're pretty much fine. Even the length of their skeletal rods doesn't look like even calcification is impacted. But certainly, the static cultures which we are using, which was just one consistent, it fluctuated a little bit, but probably less than 0.1 pH units, would give you different results than if you had sort of a feedback mechanism that could adjust the pH according to the tidal cycle or something like that, which some other researchers have been doing. Yeah. So you get a very fine sort of snapshot. And the other thing that that's important to consider is, we looked at early life stages. It doesn't mean that the later life stages are going to respond in the exact same way. So lots of other variables and future experiments to do.
The salmon, you said that it might be a virus infecting them because you saw the new genes getting upregulated, and the ones that had them not so high. Does that mean that their offspring are likely to also show the same survivorship?
So that's an excellent question. We don't know. The working hypothesis was that there was some virus in the marine environment that these salmon were picking up along their way before getting to the river, and of course they were either not making it to the river or they were dying. They were dying on the journey. I'm not familiar with how much that has progressed from that point. So the data I showed you was sort of a preliminary study, and they after I left, they did a much larger scale version of it. That paper is studied or published in Science, and it shows these big expression differences between the survivors and the mortalities, and they hypothesized that it was a virus. So based on just looking at the gene expression patterns. So that hypothesis is certainly possible, but there's really not that much evidence for it. It could be a number of things, and so they haven't identified the virus, and they don't know if it gets passed on to their offspring, for example. Yeah.
So for the salmon one, did you sample any of the ones that had already successfully made it that you hadn't caught before? Because how are you sure that that's differences in expression due to taking the journey as opposed to those ones that made it that are handling the stress?
Yeah. So we don't know the causative factor that caused the salmon to not be successful migrants. So we don't know the causative factor. We just know, or we can be fairly certain, that there's this difference in expression between the ones that made it and ones that didn't make it. So another follow-up studies that has been done since I left is that they've done exactly what you said. They've sampled salmon at various points, so that in the ocean, in the river mouth, and then various points all the way up through migration, and they can look at how the physiology changes along that line. But if you do it that way, then you can't really separate the survivors from the mortalities because you're only always sampling the survivors, right? So that biotelemetry is really the only way we can look at survivors versus mortalities, and it places some limitations on experimental design. Yeah, but we've tried to get at that question.